Right, so now it's time to take a look at translation, which is where we're going to use RNA as well as ribosomes and a few other things to actually manufacture some proteins. So the first thing we need to take a look at here in this section is the genetic code. And so you can think of this section as being kind of like the secret decoder ring of the genetic world, in that everything we need to go from RNA, or what was initially DNA, to protein is on this table. And so the way that this works is if you take a look at a section of RNA, RNA is broken up into three-letter, what essentially are words, which then are translated into amino acids, which of course are the building blocks that you need to make proteins. So for example, if you have a stretch of mRNA that reads UUU, remember RNA has the bases of A, C, G, and U. If you have a stretch of RNA that reads UUU, a ribosome would then know to plug phenylalanine in for that particular amino acid. And then if you had a section which read UCC, the ribosome would know to plug in serine to that part of the protein. So this is actually fairly straightforward. There are a total of 4 different bases, 4 different kinds of nucleotides in RNA. There are 20 different amino acids. And so since we have 3-letter sequences only that we're working with, there's actually 64 possible 3-letter sequences we can use to code for amino acids. Now, one of them is a start sequence. And in methionine, the start sequence, this one's special because all proteins, at least initially, start off with what's called an AUG start codon. In the RNA language and DNA language, codons are 3-letter sequences of bases or nucleotides. There are also 3 stop sequences, which correspond to a place where a ribosome is going to know to stop translation. So that leaves us with a grand total of 60 other different codons, which are used to code for the other 19 different amino acids. So methionine is one amino acid, there's 19 other ones. We've got 60 other codons we can use to code for those other 19 different amino acids. And since there's 19 amino acids with 60 possible codons, we know that on average there's almost 3 codons per amino acid. There's actually a fair degree of redundancy or fail-safe built in here, that if you actually have a wrong letter, for example, if RNA was supposed to read CGU, but there was a mutation that changed to CGC, you actually would still get the same amino acid, arginine, and that actually would lead to the same protein being produced that was supposed to be produced in the first place, which is kind of cool. So the other molecules here, the helper molecules are going to need to do translation, are not only mRNA, which is the message coming out of the nucleus, which signifies what DNA instructions actually read, but also tRNA. Transfer RNA molecules are used to carry amino acids from the cytoplasm to the ribosome, where they're actually assembled together to make a protein. And then the ribosome itself, which is the protein synthesis machinery of the cell, and we'll take a look at how those work here very shortly as well. So tRNA molecules carry the amino acids to the ribosome. The ribosome actually puts the amino acids together to form the finished protein. Okay, so in the case of tRNA molecules, there are 2 very important parts to them. tRNA molecules are made up of a single strand of RNA. It's actually folded over itself, so it looks double-stranded. Even though technically it is officially single-stranded, it looks as though it should be double-stranded. There is what's called an acceptor stem. That's the part of the tRNA where the amino acid sits on its journey from the cytoplasm to the ribosome. And there's also what's called an anticodon. And the anticodon is a 3-letter sequence of bases that correspond to a particular stretch or codon of mRNA. All right, so if a stretch of mRNA reads AAU, then the anticodon for that particular tRNA would read UUA. So it's got the opposite bases as compared to the codon that it's sort of matched up with. So the codon is in the mRNA, the anticodon is in the tRNA, and they match up, they're complementary, they've got corresponding opposite bases relative to each other. Okay, so for our tRNA molecule, it looks something like this. We have an acceptor stem region where methionine in this case sits. We have a strand of tRNA which kind of folds over back and forth over itself. Down here with the anticodon, which in this case is UAC. UAC anticodon would correspond to AUG codon in a piece of mRNA. And so that's the rest of this tRNA molecule, is just the rest of the strand, which kind of folds down and around and eventually ends up right here. Right, the ribosomes are made up of 2 parts. There is a small ribosomal subunit, and there's a large ribosomal subunit. Okay, so the small subunit is made up of approximately 1,900 bases of rRNA and roughly 33 different proteins. So if you think all the way back to chapter 3 when we talked about what ribosomes are of course protein synthesis machinery, but they are made out of rRNA and proteins together in one sort of relatively large complex molecule. The large ribosomal subunit down here is quite a bit bigger. It's almost 5,100 RNA bases, rRNA, and about 49 proteins, so it's a much larger part of the overall ribosomal functional unit. All right, so as we mentioned before, there are 3 steps to transcription and translation, and eventually in the next chapter, as we'll see, replication. And they all have the same names. There's going to be initiation, there's going to be elongation, and there's going to be termination. So the actual specifics are different, but the names of the steps are pretty much the same thing, if not the same thing. Right now, what we're not going to be able to do in this lecture is to work through an example of translation like I do on the board in class, because it's just not conducive in this format to do so. But we'll still try to describe the process as best we can. Okay? So in the case of initiation, the first thing that happens with initiation of translation is the mRNA molecule, which has come out of the nucleus. It's been transcribed, it's been processed, it's got a cap, it's got a tail, it's been spliced. That mRNA comes out of the nucleus and is going to bond to the small ribosomal subunit. Okay, at that point, we're going to have the mRNA molecule sliding through the small ribosomal subunit until the AUG start codon is in the first binding site of the tRNA molecule. So the first binding site in the tRNA molecule, in the ribosome has to bond to the AUG start codon, which is on the mRNA. At that point, once the AUG start codon in the mRNA is on the first binding site of the small ribosomal subunit, then we're going to see our first tRNA coming in, carrying methionine to the ribosome and dropping it off. Okay, so at this point, we have what's called an initiation complex. We have the mRNA, we have the small subunit, and we now also have a tRNA molecule carrying methionine, in the case of this example, in to be dropped off. Okay, so at that point, we're going to have the large ribosomal subunit come in and join the complex, and that's going to be more or less the end of the initiation of translation. So mRNA joins up with small subunit, it sort of scans along until it finds the AUG start codon, the tRNA comes in carrying the appropriate amino acid to that codon, and then eventually the large subunit comes in, and that's going to be the end of the initiation stage of translation. Okay, so that's kind of our first step. Now, the second step is elongation. And elongation is the part that takes the most amount of time, and elongation is the part of the process where we're actually going to build a protein. Okay, so now we're going to have our second tRNA coming in and bonding to the second binding site on the ribosome. So whichever tRNA has the anticodon, which is complementary to the codon in mRNA, it's going to carry in its corresponding amino acid to the ribosome and get ready for a transfer to occur. Okay, so at that point, we're going to have a peptide bond, a bond happening between the carboxyl group of one amino acid and the amino group of the next amino acid, forming between, in this case, methionine, which was our first amino acid, and whatever our second amino acid was based on the codon in the mRNA molecule. Okay, so we have a peptide bond forming between 2 amino acids. At this point, we actually have, in the case of this example, the world's smallest protein because it's only 2 amino acids in size. So it's very, very small, very, very simple at this point in time, but it's a starting point to build bigger and more complex proteins. Right, so at that point, the first amino acid is left behind at the ribosome with the other amino acid, to which it is now bound. And the empty tRNA molecule, the one that brought in this case, this example, methionine, is going to be kicked off the ribosome back into the cytoplasm where it can pick up, in this example at least, another methionine and bring it back to the ribosome should another one be called for. So at that point, the entire ribosome is going to slide down the mRNA by one codon. The tRNA that was left behind now with 2 amino acids is going to be in the first binding spot on the ribosomal subunits, and we're now going to bring in the next tRNA molecule carrying the next appropriate amino acid complementary to the mRNA codon that we see in the mRNA. So the tRNA with the complementary anticodon comes in, matches up with the codon in the mRNA, and we're going to go ahead now and rinse and repeat. So the first 2 amino acids that we had bound together are going to transfer to this third one. We're going to kick out the second tRNA that came in, and then we're going to shift down by a single codon. And so that keeps occurring over and over and over and over again until we reach the end of that particular piece of mRNA. This keeps happening over and over and over again. So we create a peptide bond, we kick out a tRNA, we bring in another tRNA, create a peptide bond, kick out the old one, and so forth. So that's essentially what elongation is. The last step here is going to be termination, and with termination, now we're going to have the stop codon on mRNA, and there are 3 of those, 3 different stop codons. As soon as one of them shows up in the second spot on the small ribosomal subunit, that's going to lead to termination. And so there's going to be a series of proteins that come in here. These proteins are going to bond to the ribosome. You're going to break it up into parts. So the small subunit goes one direction, the large subunit goes some other direction. The finished protein is released. tRNAs are released as well. And mRNA is then available to be transcribed or translated again. at least one more time, if not more than one more time. So everything gets broken apart. The ribosomal subunits go one way, tRNAs go one way, mRNA goes back to be translated again, the protein's done, that's going to go off potentially, say, to the Golgi apparatus to get finished off. Everything is now broken apart at this point in time. Right? So that's going to be termination. Okay, so the last section that we're going to see here, I think, is going to be on protein synthesis and regulation of that process, and then also a look at what happens when mutations occur and the kinds of effects that mutations can actually have on the physical appearance or phenotype of an organism that has that particular mutation.